Welcome back, everyone, for another chapter of biology. This chapter is going to be dedicated to cell structure. So we're really going to start diving in to what cell we've talked about before, what cells are made of. Now we're actually going to see what an intact living cell actually looks like. This first video is going to discuss cell theory and the role that microscopy and microscopes have played in us being able to observe cells. It's going to be good to have some historical background and context before we really start diving into what cells actually look like and what different parts of cells do. Okay, so let's recap just a little bit from some of our previous discussions just to make sure we haven't forgotten anything. The cell is the smallest unit of life, so life is entirely defined on things that are made of cells. All organisms are made of cells, okay? So we talked about how there are two major types of cells. So we've got prokaryotic cells and eukaryotic cells, and that is the basis for how we classify domains. So we've got prokaryotes, eukaryotes, okay? So you'll have to remember that some of the differences that we're talking about between prokaryotic cells and eukaryotic cells are based on microscopic observations, which obviously are going to require the use of a microscope in order to be able to see those things. Hence the reason we're having a microscopy discussion in this video. So a microscope is an instrument that greatly magnifies the apparent size of an object relative to its actual size. So you don't need me to explain that to you. You probably know what a microscope is. You could probably look at one and tell that, hey, that's a microscope. You have an idea of, okay, it's going to make things appear a lot bigger. But there are some subtleties to microscopy and different types of microscopy that we can use for a number of different applications that are going to be good things for you to know. Okay, so let's start off with a little bit of historical context. Where did the first microscope actually come from? The first single-lens microscope was invented by a man named Anton von Leeuwenhoek. So this was mid to late 17th century, so we're talking kind of the 1600s, 1650 to 1690, that sort of range. He used his microscope invention to observe bacteria and paramecia. One of those is a prokaryote, the other is a eukaryote. Now, this was a little early for us to really start laying all the important groundworks that biology and cell biology are made of, but this was obviously an important watershed moment. The term cell was actually not coined until 1665, which is actually a little bit before Leeuwenhoek did his work. This was more speculation by Robert Hooke than anything else. So he speculated that living organisms are made of little individual units called cells, even if we didn't exactly have the microscopic evidence needed for that at that point. So this was actually great, though, because we actually were able to confirm this with more detailed studies of cells using more advanced microscopy as early as the 1830s, kind of the early 19th century thereabouts. Actual cell theory, which includes all the tenets and all the unifying principles that really bring all these different organisms together, were laid out by two gentlemen named Schleiden and Schwann. The main thing we want to focus on here are the tenets of this theory. Number one, all organisms are made of cells. Number two, all cells come from already existing cells, meaning there has to be some kind of reproductive process in order for new cells to be made. In other words, there is no spontaneous generation. Cells don't just magically pop up out of nowhere. The only way to get new cells is to get a division or cell division process coming from cells that already exist. And finally, cells are the basic units of structure and function for all organisms. You cannot talk about a living thing without talking about the function of its individual cells. Okay, so a lot of the tenets of cell theory are things that we have already discussed back in Chapter 1, so let's start talking about cell size. So, for the most part, cells are going to be much too small for us to be able to see without the use of a microscope, but even considering that, cells do vary in size just a little bit. If you look at this chart here, you can actually see kind of a very wide range of different sizes of different structures that are important in biology. So something like a blue whale, which is one of the largest organisms that exist on planet Earth, this thing is between 10 and 100 meters in length. So that is something that is very, very big. Humans tend to be roughly about 1.5 meters to maybe 2 meters for someone who's very, very tall. So we're clearly much smaller than a blue whale. And then as you kind of go down and down and down, you start to look at individual cells. So plant and animal cells are going to be somewhere between 10 and 100 micrometers in length. A human egg is going to be much, much, much bigger. A frog egg is actually a rare example of a single cell that you can see without a microscope. And then as you kind of go down and down and down, and you start looking at individual proteins, small molecules like amino acids, and then individual atoms, there's going to be a limit to what you can actually use microscopy for. We can say with certainty the one thing here that you definitely cannot see without even with a microscope is an atom. There's just no way to see individual atoms. They are way too small, even for an electron microscope, which is capable of seeing very large molecules like proteins. So most cells are going to generally range between 1 micrometer in length, that's 1 times 10 to the minus 6 meters, and then 100 micrometers in diameter. You might also sometimes see that unit of measure called a micron. So you'll notice that there's not really a very wide range of cell sizes. And we're going to find out on the next slide that cells have evolutionarily developed to be in this narrow range of sizes because it helps to maximize their surface area to volume ratio. So the reason why that is important, as we shall see with a demonstration on the next slide, cells routinely have to transport molecules and other materials across their plasma membrane. These transport processes become extremely, extremely, extremely slow and inefficient if that ratio is too low, which is generally what happens as cells get bigger and bigger and bigger. So there's clearly a reason why cells have to be as small as they actually are. Speaking of this surface area to volume relationship, the idea here is that if a cell is too big, meaning if it's big enough that we can actually see it, that surface area to volume ratio is going to be small, and that's going to mean that transport processes and metabolic processes will be so slow that they won't be able to support life and living functions. So if you look at this particular example here, if we look at one single cubic object that is actually a single very large object, it has a total surface area of 96 square centimeters. Okay, so if you add up the surface area of each face of the cube, each face being a 4 by 4 square, 4 times 4 is 16, times the 6 faces would give you a total surface area of 96 square centimeters. Okay? The total volume of this cube is 64 cubic centimeters. That's 4 times 4 times 4. Length times width times height. So the surface area to volume ratio here would be 1.5 to 1. Okay, so 96 divided by 64. Well, look what happens to these numbers as we take that same cube and divide them up into 8 individual units or 64 individual units. You can see that the total surface area increases without changing the total volume involved. So in doing so, we drastically increase the surface area to volume ratio by taking one single large object and splitting it up into a number of smaller objects that collectively have the same volume. So the idea here is that making a cell smaller and smaller and smaller actually causes this surface area to volume ratio to go up, and in doing so makes transport processes and metabolic processes go faster. Now, you might be thinking, okay, well, if we make a cell smaller and smaller and smaller, that's going to make things more and more efficient. So why don't we just make cells extremely, extremely small? Well, think about it. Can you think of why there might be a limit as to how small a cell can actually be? We want cells to be small enough to maximize the surface area to volume ratio, but we can't make them so small that they can't contain all of the necessary molecules that are necessary for life, like DNA and proteins and things like that. So we definitely don't want cells to be too big, but there is a clear limit into how small they can actually be. You can see the same sort of thing with this figure here. when we're comparing a very small cell with a very large cell, you can see that the surface area to volume ratio is 10 times bigger for the smaller cell than it is compared to the bigger cell. Okay, with all of that in mind, that explains why cells need to be very, very small, so we can understand that because cells are small like that, we need microscopes in order to be able to look at them. So let's talk about different types of microscopes and different types of microscopy. The first one we're going to talk about is called compound light microscopy. The nice thing about this is that a compound light microscope is easy to get, cheap to buy, and effective for most general purposes. So compound light microscopes use two lenses, so they're a little different from von Leeuwenhoek's initial microscope that he made, which only had one lens. Because there are two lenses, this is going to cause any object that you look at to be inverted and flipped on both the x and the y-axis. A common thing that you will look at when you are performing a biology experiment as a biology student is often you'll look at a slide that has the letter E printed on it. So you can clearly see on the slide that the letter E looks correct, but when you look at it under the microscope, the E is going to be flipped upside down and then turned around 180 degrees compared to what it actually looks like on the slide. The way a compound light microscope is going to work is that there's going to be a light source that is usually powered from electricity. This light is going to be reflected through a mirror, and then it's going to be condensed up through your first lens, which is called the condenser lens, and then that light will then pass through your specimen, and then after the light passes through the specimen, it will go through what is called the objective lens, and then finally a set of mirrors will reflect this so that the light eventually passes through the cornea of your eye, and then an image will be formed on your retina so that you can actually see what you're looking at. The major thing that's happening here is that as the light comes from the light source and then eventually ends up on your retina, because of the way the light is being passed through these lenses, the object that you're looking at on the slide here, the specimen, is going to appear several orders of magnitude much bigger than it actually is on the slide. Compound light microscopes provide a magnification range of usually 40 times to 100 times. So the ocular lens up here provides 10x of magnification, and then the objective lenses here can be changed out. The smallest objective lens provides 4x magnification, so you multiply those lenses together to get the total magnification. If you're using the 4x objective and you multiply that times the 10x ocular, 4 times 10 gives you 40x magnification. The objective lenses can take steps up to 10x, 20x, 40x, and 100x. So if you're using the 100x objective, you can get 1,000 times magnification. You can make an object appear 1,000 times bigger than it actually is. This picture you're looking at up here of Euglena, we are using 200x magnification, so a 20x objective was used to take a peek at these. The compound light microscope also provides resolving power approximately 500 times better than the human eye. We will discuss resolution at the end of this video, so don't worry too much about that for right now. Now, one of the bad things about the compound light microscope, it's obviously very easy to use, very easy to get, and it works for most things, but there are some limitations. Some objects in a cell are just way, 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 way too small for us to be able to see even with a compound light microscope. So that's where electron microscopy comes in. So the downside of an electron microscope is that they are extremely, extremely expensive. You are very unlikely to ever use an electron microscope yourself, even as a biology student. The cheapest kind of electron microscope is going to cost somewhere in the neighborhood of $100,000, and some of the better ones and more expensive ones may cost upwards of $1 million. But they are absolutely unparalleled in terms of their effectiveness. There are two major types of electron microscopes: transmission electron microscopes, which you can see on the left, and then scanning electron microscopes you can see on the right. You don't need to be concerned too much about the difference between them, but what electron microscopes in general are going to use is that rather than a beam of light that is passed through lenses and mirrors, This is going to use a beam of electrons that are passed through a specimen, and they are focused instead of glass lenses and plastic lenses by magnetic lenses instead, and the image will be reflected onto a fluorescent screen where you can actually look at them instead. So you're not really going to need an eyepiece to look at your image here. Electron microscopes are incredible in the sense that they exceed the magnifying power of compound light microscopes. Your magnification level can get to over 100,000 times magnification. In these pictures that we're looking at here of the euglena, with the transmission electron microscope, you can actually see very specific structures inside of the cell, which was not possible just from using a compound light microscope. The scanning electron microscope here can actually provide a much clearer and crisper image much closer in than what we saw before. But one of the other downsides with electron microscopy, you cannot look at living cells with electron microscopy. Electron microscopes involve treating the specimen with certain chemicals and other sorts of things that will ultimately kill the cell. So you cannot use electron microscopes to look at living cells and living tissue. Okay, so let's talk about some terminology that are associated with microscopes. We've already discussed magnification. This is the ratio of the size of an image compared to the actual size of the object. So if your magnification level is 1000x, the size of the image that you're looking at is actually 1000 times bigger than what the object actually is. Resolution, which we mentioned before, this is the minimum distance between two objects that allows you to distinguish them as separate objects. You've probably heard this piece of terminology when thinking about things like TVs. So having resolving power means that you can have two objects that are extremely close together, but because you have a great deal of resolving power, you can actually perceive them as two separate objects. If your resolving power is not good, you could have two different objects that are very close together, but because they are so close together and because your resolving power is so limited, they look like one separate object, so you wouldn't ever know that they're two different things because they actually look like one thing. And then contrast is the difference in shading of an object compared to its background. It's possible for something to appear invisible because it is indistinguishable from the background. Having a high level of contrast allows you to actually see objects and distinguish them from the background. A good example of this is in scanning electron microscopy. You generally spray the specimen with some kind of heavy metal atoms like gold in order to act as a contrast agent for imaging. And then another example of this, which we didn't really get a chance to talk about, is that in some types of microscopy that involve lasers, you can actually use fluorescently tagged molecules, and you can actually label different components of the cell with these different colored molecules to distinguish them from a dark background. Alright, that is going to do it for this first video on microscopy and cell theory. Thanks for your attention, and I will see you next time, and we will rehash some previous discussions we've had on the difference between prokaryotes and eukaryotes. See you next time.